So welcome everybody. Welcome to this 17th edition of the Nobel Lectures at the University of Twente. My name is Gert-Jan Koster. I will be the host, and we have some fantastic speakers. We do this every year. We introduce the Nobel Prize as quickly as possible after they have been awarded, to introduce the topic and a little bit about the prize winners if possible. So it's always these 2-3 weeks after the prize has been awarded. It's a little bit hectic to find the right speakers. And again, we found some fantastic speakers for today for physics and chemistry. So I will not talk any longer. I will start with the first speaker of today, Wilfred van der Wiel. Professor van der Wiel did his PhD in Delft, but also combined with NTT in Japan. And in 2005, he joined the Mesa Plus Institute here at the University of Twente. And now is actually partly involved in developing electronics, if you like, that actually hosts AI. Please, Wilfred, the floor is yours.
Thanks a lot. Thank you. Is this working now? I think so. Yeah. Thanks a lot, Gert-Jan, for the nice introduction, and thanks for Studium Generale to knock on my door. It was not the thing I was, of course, really hoping. A phone call with someone with a Swedish accent, but getting a phone call from Studium Generale in Twente, I think, is a very good alternative and second best. To me, the task and the honor, the duty to try to explain a little bit the Nobel Prize in Physics 2024, awarded to John Hopfield, US citizen, and Geoffrey Hinton, Canada, for foundational discoveries and inventions that enable machine learning with artificial neural networks. And I thought maybe it is not a bad start to focus on that first sentence, because that's the essence, of course, of the Nobel Prize. And we are every day kind of bombarded with the word AI and all the societal implications, technological implications it has, ethical maybe even. So I thought, where do these words, machine learning and neural networks, artificial neural networks, fit in this much larger domain of artificial intelligence? So artificial intelligence and maybe more traditional data science have some overlap, as you can see, and machine learning is quite a large field that lives at the kind of cross-section of these huge fields nowadays of AI and data science. And you can do machine learning in different ways. So maybe to make clear what is machine learning, basically instead of writing out a computer program line by line, give an instruction set, okay, this is what you have to do. I remember from my basic 10 print, Wilfred, 20, go back to 10, and then you can just all the time watch your own name for the rest of the Sunday afternoon. So that's let's say, program-based computing. Machine learning is basically trying to start from scratch with a structureless model and see if you can train it, if you can learn it by meaning of input data and a learning algorithm, so that at the end of the day also you have an analytical model that can be doing a certain job for example, pattern recognition or speech recognition or something like this. So the philosophy is quite different from traditional computer programs. And it's probably also interesting that in the beginning of AI, also people try to really go from logical rules, rule-based approaches to do things like pattern recognition. And it's especially Hopfield who try to break from that tradition and move more towards brain-inspired approaches. Then within this machine learning, there is then specifically neural networks inspired by the brain. I'll get back to that, which is a subcategory where you really look at models that are consisting of nodes, of weights, synapses, and build your model based on these building blocks. So just to give you a little bit an idea where we are playing, what is the playground. So we have machine learning and then neural networks here. Okay, so what are artificial neural networks? I will maybe use also just neural networks, but then I always refer to basically the kind of abstractions based on biological neurons. And here you see a cartoon of what we now understand about the structure of biological neurons. There are actually many kinds of neurons, maybe there's like 100 different kinds of neurons, but this is a typical a neuron as we have in our brain with a cell body and a soma. It has many dendritic arms that receive signals, spikes, electrical spikes from neighboring neurons. One neuron can be interconnected to up to 10,000 other neurons. So this interconnectivity is super high, and that also gives actually the brain its large computational power, this parallelism. And then the cell body integrates, basically sums up all these signals that it receives from other neurons, and then if some certain threshold is reached, it spikes. There is an action potential that then goes along its exit channel, the axon, which again is then connected to a whole set of other neurons. Now, what is happening at the interface between these dendritic ends and other neurons is very interesting, because there is a plastic element, which with plastic I don't mean it's made of PVC or PET or something, but what I mean is that it's able to actually change its actually structure, its hardware, so it can actually strengthen or weaken or completely be disconnected, and that's the synapse. And the synapse is a very interesting cleft in between ends of these dendritic ends of neurons, where you have a kind of collaboration of electrical signals and also chemical messengers, neurotransmitters, that finally make that these signals move from one neuron to the other. And it was already in the 40s when Donald Hebb came with the proposal that there should be a kind of connection to the activity in these synapses and the actual strength. And the slogan that made it was "cells that fire together, wire together." So actually, if there is a kind of causal relationship between the spiking patterns of interconnected neurons, you actually strengthen these connections. Another nice slogan is use it or lose it. Yeah? So if you don't use your brain enough, then after some time, maybe there is not too much left of it. So this is a kind of interesting principle, which you could also see as associative, kind of form of associative learning. So if there are events happening that can be associated, it leads to learning. Another interesting connotation is that it is a local learning rule. So it means that you basically only need a very local information of interconnected neurons to base this learning on. So you don't need a kind of external supervisor or conductor or software to let that learning happen. So a local learning rule. Okay, so now if we go to the abstraction, you could say, well, this is a kind of still the biological neuron. By the way, again, already in the 40s, there were people who tried to make up these models for abstracting these single neurons, these nodes. So you have the cell body again, you have these incoming signals interconnected with synapses, and if you now really go one step further in abstraction and you try to make a model out of this, you can write it as follows. So you have this summation of the incoming signals. The incoming signals here, they are just multiplied by weight factors. So these could be your kind of synaptic strengths. Sum them up, and then you have at some moment a nonlinear event. So you hit some threshold. This is, in biological terms, your action potential. And in this model, simply once you meet a threshold, then a function, a nonlinear function, goes from 0 to 1, or from -1 to 1, for example. So this is really a very basic building block of a lot of neural networks, this element. And if you now start to interconnect these in a way that also in a biological neural network, there are all these interconnects, then you get something that is an artificial neural network. And normally speaking, in order to basically kind of don't lose our track of what's going on, we order these artificial neurons in layers. So we have an input layer, and that is then connected to the next layer. And finally, you can have many intermediate layers. We call them hidden layers. And then finally, a layer that is connected to the outside world, which is the output layer. And in this case, you see that basically all the neurons from one layer are connected to the next one. And the information in this case always travels forward, so from left to right. This is not necessarily the case. We will see that Hopfield and Boltzmann networks are actually not according to this feed forward, but many of the modern deep neural nets are of this kind, feed forward, and in this case also fully connected. Maybe you have heard about deep learning. So deep learning is, very simply put, many of these hidden layers between input layer and output layer. So then, what is now the game that people are playing in this machine learning with neural nets? Well, for example, you would like to train such a spaghetti of neurons and interconnects to solve this pattern recognition problem for you. So you would, for example, encode these images, I don't know, 256 by 256 pixels, for example. You encode the whole image, maybe also in red and green and blue, so you have even more inputs. So you have thousands and thousands of inputs. You have maybe millions, if not billions, of weights, and you start tweaking these weights till at the end of the day, when you basically present an image of this dog to this network, miraculously, it will give the right answer, dog. Not cat, not duck. And the only way you can do that is by training this network with thousands and thousands of examples of dogs. And you label them and you say, well, if you make an error, you have to change a little bit these weights. And actually, Hinton was one of the main players who found a very elegant mathematical trick called backpropagation to optimize the weights of these huge networks. He didn't get the Nobel Prize for this, so this was just a kind of little Friday afternoon side job that he did. Extremely important backpropagation to train efficiently these large neural networks and use an error function or a cost function and based on that backpropagate errors to change all these weights in this network. So that is a kind of the art of machine learning, deep learning. It's a big black box. So you have maybe no idea where you have to put all these certain weights. The only thing that finally is the proof of the pudding is the eating, namely right labeling of these images. We go now to the first Nobel Prize. One half of the Nobel Prize goes to John Hopfield. He's 91 years old, so that makes me very optimistic still that I still have some time left to maybe to have a chance. The Hopfield network is also a kind of artificial neural network. But you can see it looks quite different from the ones that I showed you before. This, of course, is schematic. But what is, let's say, fundamentally different is that this is a so-called recurrent neural network. That means that information is not only going one way, but it can actually also loop back. So an output of a neuron can actually at some moment be also fed back to its input. And that is kind of more biological plausible also. I think also in our brain this is happening. So that's a big difference. And it's also slightly different from the task that I explained to you earlier. So let's say image classification that I explained to you before. This is a kind of model system for an associative memory. So what you can do in a nutshell is you can basically program the network and encode a couple of memories in this network. And then once you confront the network with that image or pattern that you have encoded, it can recognize it. Not only that, even if you come with a slightly distorted version of that pattern, so let's say we take an image of Peter Timmerman, for example, and we encode it in this network. Even if I come with a very old, noisy, little bit wrinkled version of it, this network could be quite well able to recover the original image, so giving the answer basically, this is Peter Timmerman, by showing the non-distorted version of Peter. That's a comforting idea, I think. Yes, yeah. So it stores and also can reconstruct images or patterns. And now the interesting thing is, and that's maybe the clue why this has anything to do with physics and why we should be not really surprised that there is a physics Nobel Prize for AI, because this is a model that you can describe very elegantly and nicely in terms of physical model. So I'm going now a little bit. This is for the people who really would like to know a little bit the fine details. But even if you lose track here, we'll be back on the main road very soon. So don't worry. But what basically the definition of this network is that you have these neurons. And these neurons can be on or off. And on we call 1, and off we call -1. And no, I'll do it the old-fashioned way. Every neuron is connected to all other neurons, except to itself, via a weight. So there is this weight factor that I introduced to you earlier. And there is also a threshold value for every neuron. So that is a certain a value that you will compare at some moment the inputs to. I introduced this threshold value also to you earlier when I had this nonlinear step function. And now the interesting thing is that you can describe the state of the entire network by a kind of quasi-energy. Of course, a mathematical model, which it is. It doesn't make really sense to talk about energy because it's an abstraction, but let's call it energy, and we'll see for a moment why that is. And now this is the update rule, that you change the state of every individual node based on basically the state of all the other ones in the network weighted by these weight factors. And then this threshold value comes in. So dependent on what all your brothers and sisters are, you change this state. And the interesting thing is now you can define this energy in terms of the states of the individual neurons, these S's here, and these weight factors. And I will come back to this energy expression because this is an energy expression we know very well from physics. There's a question.
Yeah. WIT should always be symmetric?
In the case of the Hopfield, it's always symmetric. Yeah, that's a good question. Yeah, so both ways the same. It's correct. Yeah. Okay. So the analogy with physics is that these nodes, that is these artificial neurons, actually behave as if they were little magnets, or as we physicists like to call them, tiny magnets, or small bar magnets. And the weights between these small magnets are a kind of magnetic interactions. And now, as in magnetism, where you have basically many atomic scale little magnets, which come from the internal structure of individual atoms, the interactions lead finally to a kind of global behavior, like ferromagnetism. So local interactions give rise to global behavior. Emergence is also a kind of popular term that you hear in this context. So here you see an example. If you have many of these small magnets or magnetic moments, if the coupling between them is not so strong, you basically have a chaotic system, no order. We call this a paramagnet. But if there is a coupling that tends to align these neighboring magnets, so just nearest neighbor interaction, then you arrive at a highly ordered system which is ferromagnetic. And the energy of such a system can be, in a simple model, relatively simple model, to be described by the Ising model for a magnetic state. And I don't want you to really go to the fundamental of this equation, but just compare it to the equation for the Hopfield network. So this is a description of the physical world, and this is a description of a mathematical abstraction. And even if you have never seen these formulas, and maybe even if you have never seen these symbols, I think our pattern recognition abilities are good enough to say that they are very similar. And they are actually in essence. So basically, the Hopfield network is a kind of one-to-one projection of a magnetic system onto the world of neural networks. And it goes further, because the update rule that I explained to you earlier, so basically the rule, the kind of recipe I gave, whether a neuron should change for a 1 from 1 to -1 or -1 to 1, is one-to-one connected to this energy. The way that these individual neurons are changing their state are always going in the direction of lower or at least equal energy. So like a magnetic system tries to order its state in order to minimize its overall energy, also this Hopfield network, if you disturb it, it always moves into a state of lower energy, where this energy is now this mathematical energy. So for example, you start in a certain state of this network, you kind of disturb it, and then it moves to a lower energy state. So the mathematical construct is such that by nature, by construction, if you shake up this model, it will move to a minimum. So for example, if you have your Hopfield network, which has been trained by these weights to have a minimum state that corresponds with 1, or 1, and -1, 1, -1, 1. So this is really a low energy state that would correspond to a minimum here. If you now give it an input that is nearly the same as this minimum, but not quite, because it differs by this number, then actually this update rule will push it basically into this state. So it is what mathematicians call an attractor. So you know maybe attraction in a completely different context, but mathematicians, when they talk about attraction, they mean this. And to make it even a bit more visual, if you look in a landscape, this energy landscape given by this equation, you could have regions of attraction that correspond with a local energy minimum. So, and in this case, you could have here the image of Albert Einstein corresponding with this local minimum. And in this local minimum, you have the picture of Hans Hilgekamp is excluded from participation, but yes, excellent, yeah. He got 2 Nobel Prizes, by the way. So there is baas boven baas, as we say in Dutch. Although Einstein, as you probably know, was also not a little guy in the world of physics. Anyways, so yes. Okay, perfect. Yeah. So these attractors. Now comes the way how these models work. You come, for example, with a heavily distorted version of these pictures, and the network will actually automatically condense or be attracted to these possible solutions. So you start with a completely messed up picture, and the output of the network gives you the result. Okay, let me move to Geoffrey Hinton, born in Wimbledon, that you may also have heard of in a completely different context. And Geoffrey Hinton built built on the work of the Hopfield model. And that's why I put quite a lot of effort in explaining the Hopfield model in detail, because if you know the Hopfield model, you can basically extend that to a system where now the states of the individual neurons are not deterministically determined by this update rule, so whether they are -1 or 1, but the states of the individual neurons, so whether they are 0 or 1 or -1 or 1, depending on your definition, depends now on a statistical rule. They have a kind of statistics, a probability associated to them. And that's where the word Boltzmann comes from, because again, these guys borrowed ideas from physics, namely the Boltzmann distribution. So in this case, we also have an energy function that again looks very similar to the Hopfield network. So we have again the states of these neurons, weights, thresholds, but now the probability that you are in a certain state, state S, is given by this expression. This probability for a state is an exponent, energy, and temperature. And this is basically the distribution that we know from Ludwig Boltzmann, which says as much as the higher the energy of a certain state, of a gas is, of a system of molecules, the higher the energy is, the lower the probability is to occupy that state. I can't go into all the details, but what is of course striking, that already starting from a model that is largely based on physics, Hinton adds another physical flavor to it, namely statistics. And by inserting this statistics in this model, the neural network can do much more than just regenerating known pictures. It can actually generate also new data, and it can structure data, it can find new patterns, because this probabilistic, very difficult word, probabilistic nature of the network helps to explore also different possibilities. So just to compare, Hopfield is reproductive, so it retrieves stored patterns, but the Boltzmann machine can also generate. It's generative. And generative you also know from ChatGPT. There is generative also there. It's a generative, a pre-programmed, pre-trained transformer. So the fact that also these language models can generate new stuff based on your trained input, that is also, it's a very, very different model, transformers and Boltzmann model, but its generative is similar in that. Hopfield is still deterministic, but Boltzmann machines are probabilistic. So they come with this Boltzmann statistics. And there are some more details, which I will skip for now. And a very important contribution again from Geoffrey Hinton is that he came up with an idea to go from this Boltzmann machine to a more simpler case, which is called the restricted Boltzmann machine, which became a kind of main building block for training these deep neural nets. So with having this so-called restrictive Boltzmann machine, it was first of all much easier to train a Boltzmann machine, but also enabling again this huge explosion of deep learning. So let me kind of wrap up with discussing very briefly the impact of the work of Hopfield and Hinton. I think, and they have been called on many occasions like the kind of the godfathers of machine learning, and I think that is not exaggerated. Their ideas in one way or the other are really widely used in image recognition, LLMs, these large language models, all kind of AI applications, and they are now also used for solving problems in physics. So you can use neural networks as a kind of digital clones of physical systems. So you don't need actually to build these systems, but you can simulate them. You can do quantum mechanical problems, and you can, for example, also study phase transitions. So here you see work that was actually cited by the Nobel Prize Committee of the group of Menno Bogdam, sitting here in the audience, which I'm very scared of because he knows these things extremely well. And some work from our group where we actually also built a neural network clone of a nano-electronic device and use that to learn and to understand better. But there are challenges, because all this beautiful, nice, flabbergasting AI consumes a lot of energy. And here you just see how the number of computing steps is exploding thanks to these developments of these two gentlemen. You can do much more, but AI already uses the amount of energy of a small country. And by 2030, maybe about 20% of all electricity worldwide is being used for ICT, and a large part of that is AI related. And even if you go further, maybe by 2030, the total amount of world production of energy goes to ICT, which is of course ridiculous, if we would go in the same pace and try to realize these AI models in the present day hardware. So I would like to finish this talk with basically closing the circle. Hinton and Hopfield have basically borrowed ideas, principles from physics to build their AI models on. Why don't we go back with our models to physics and see if we can also use for the hardware physics to incorporate these models? And can we indeed exploit all these many body interactions, all this richness, complexity, these energy landscapes that physical systems naturally provide for implementing these AI and machine learning applications in a much more resource-efficient way than by brute force computing in data centers as we do today. And with that, I thank you for your attention.
Thank you, Wilfred. I'm sure there's questions, so there's plenty of time. So please raise your hand and we will bring a microphone to you. I will bring a microphone to you. Yes, please.
I would like to ask, you have an analogon with physics. Have they also been trying somebody to have an analogon with chemistry, in the sense that if you look to Gibbs energy, it will also always go to a minimum? And also Boltzmann started first because of the entropy. He started his calculations on statistics.
Yeah. Yeah, so it's the real chemist is just leaving the room. But I mean, of course, yes. Also, these principles of Boltzmann are later on picked up by Gibbs, and I think the present-day expression way that we express it is due to Gibbs. So many of these, I think, statistical mechanics principles, Gibbs energy, free energy, are one-to-one also applicable. I don't know too much about the field to give you really a pinpointed answer there. But definitely these descriptions play a role. In general, also, if you about this last part, can you use also chemistry for implementing neural networks? There has been just, I think, last month a paper by Wiel. It's a pity that David has just left, because it's his former PhD student, where he actually used chemical reaction networks, formaldehyde reactions, big mass of coupled reactions to simulate also neural networks. So when I say physics, yeah, I'm a bit preoccupied with physics because I'm a physicist by education, but it definitely also includes chemistry and everything material science that's available. Yeah.
Okay. Thanks for the nice talk. I have a question about these states. So at some point you said, like, you can be in a certain state because of the weights of the learning of the network. Can you also, once you have your answer, go back to what kind of things have the highest weight and explain why the network chose a certain result? Because I think currently we are making these systems, and then sometimes we don't know anymore what's happening and why it's happening.
Yeah.
Would there be a possibility to actually look back into why is this happening and what is the highest weight and why do we end up with a certain result?
Yeah. Yeah, this is a very, very hot debate in AI in general, which is referred to as explainability or transparency. And you can imagine the more and more prominent role these models get in society. Think about financial or think about health. People want to actually be able to explain why the model is telling them to do this or that, right? So let's say in the traditional picture, these models are black box monsters. So you have really no clue why the weights are what they are, other than knowing that a posteriori they do the job. What I have been told is that nowadays you can actually also learn models to be explainable. And I don't know the details of that, but there is a huge effort on making neural networks more transparent and explainable, especially exactly for the reason you were asking about, because there is too much in society dependent on decisions, and there is things like liability, and so this is a hot field indeed. Yeah.
Are there more questions? Yeah, please.
I had a question about the comparison between the Einstein picture and the other picture. Because in that model, would you be able to only have two options? It would be, or would there be a third option that would be, for example, it's none of the pictures?
Good point. Yeah. So these networks have a kind of finite capacity. So based on, let's say, simply put, on the amount of nodes, there is a kind of maximum amount of patterns you can store. And they are fixed. So if if I would come with my picture, it would be lost. It wouldn't be Einstein, it wouldn't be Bardeen. Maybe if there is too much noise, it will give a mistake. But in the case of Hopfield, if you haven't encoded this memory beforehand, you cannot memorize it. It's like in your case, if you haven't been on holiday to Barbados, you can't remember this holiday in Barbados in this way, kind of like that. Yeah. Maybe you can imagine it, but then it's another model.
Please.
Thank you for your talk, first of all. And I would like to correct my mistake. The second picture is John Bardeen, not John von Neumann. I shouted John von Neumann, but like,
Okay,
I got my mistake. So.
It's. I mean, von Neumann is actually also a very, very nice person. If you actually, for this Studium Generale public, if you ever have some free moment to read *The Man from the Future*, a book about the life of von Neumann, it's really very much recommendable. Because and it was recommended to me by an economist, which I found very, very strange, because we physicists now, and of course the computer scientists, always talk about von Neumann. And then this economist said, you have to read this book about von Neumann, because he's really the hero of our field. And I said, how come? You are an economist, right? He said, yes, because he had really laid the foundations for game theory and all these things. So it's amazing. So the man from the future, but yeah, here it's Bardeen.
Yeah. My question is about, like, as you also did, physicist has effect on apparently computer science, apparently on economics as well. What do you think that this prize will change the people's view for physicists or computer scientists or other people? Like,
and this, you mean this Nobel Prize?
This Nobel Prize, yeah.
How it will change?
What's your personal view about that? Because like it attracted too many debates. Like, is it physics, or like they said that computer science is now physics, like AI is now physics, or they said that physics is now AI. Or
Personally, I find it a little bit meaningless discussion, because we have at some moment decided pretty arbitrarily to say, okay, this is physics and this is chemistry. And then at some moment saying, okay, but then this is computer science. Oh, no, but this is AI. I mean, I don't care, to be honest. I think what these guys have done is great. I think without exaggerating, they could have easily earned each of them another Nobel Prize for other great stuff. So I'm extremely happy. I think sometimes you can argue, did this person really deserve it or not? But in this case, well.
If they are young, they can also win a Fields medal as well.
Yeah, but you have to be 40, I think. So I already gave up that one. But yeah. If you allow me, Chairman, there was in Delft, I had a professor, and once at a lecture, he asked, is this physics? Or are you just like Taylor, making trousers with 3 legs, waiting for someone to buy them? So sometimes, yeah, this may have been in the beginning something like trousers with 3 legs, and then later on you find out that actually people are exactly looking for those trousers with 3 legs. So yeah, it's a little bit.
On that note, I think we should complete this. Thank you, Wilfred, for this wonderful explanation. And let's move on to the prize in chemistry, which will be introduced by Halima Mouhib from the Free University of Amsterdam. Halima did her PhD in Aachen, and then after a few postdocs, she joined the faculty of the Vrije Universiteit, Free University, in 2022. And she's going to explain us all about protein folding and also the role of AI in that process. So in that sense, these 2 lectures should be quite connected. And we are very eager to hear all about the prize in chemistry. The floor is yours.
Okay. Yeah, good evening. It's a pleasure to be here, and thank you for the invitation to talk a little bit about the Nobel Prize in Chemistry, this year's Nobel Prize. So this year's Nobel Prize for Chemistry was handed out for protein structure prediction and computational protein design. And there is, of course, this immediate link to the talk that we have just seen now, because all of these works are basically founded on neural networks and deep learning. So I do not know any of the Nobel Prize winners in person, but I am aware of their work also, specifically the ones for protein structure predictions, namely AlphaFold. And so there are two parts: computational protein design and protein structure prediction. And I'm going to mainly focus on the protein structure prediction part and give a kind of a short insight into the computational protein design. Now, the three winners are David Baker, Demis, Hassabis, and John Jumper. And I've here highlighted their background. Okay, so this is really interdisciplinary research and quite challenging process in biology. We see also in their background, so this is as far as I could find on the internet, is from biology with a PhD in biochemistry. Demis Hassabis from computer science and did a PhD in computational neuroscience, so more on the applications of the biological part. And John Jumper has a background in physics, theoretical chemistry, and then did a PhD in theoretical chemistry. So it's also a mixed background in the end, and then their teams that were working on the different projects, also strongly interdisciplinary. So, might be easier. So what I would like to talk about now is protein folding and protein structure prediction directly from sequence. What do you know and what can we do about it? And then also about AlphaFold, which is the model at the moment for protein structure prediction, how it has impacted the field, and also how it got there. So what were the ingredients that were really needed to be able to get AlphaFold running and that we can actually use it now? And then, of course, yes, the impact, what are the next steps, what are the opportunities and challenges that come with these kind of models. So if you want to understand the protein folding and also the folding problem and also computational protein structure prediction, this kind of goes back to the central dogma of molecular biology. So, there is DNA, transcription to RNA, and then there's the translation to protein. And what is interesting here is the three-dimensional structure of the protein. Why are we interested in the structure? Simply because the structure is directly linked to the protein function. That means that what is the protein doing at the biological level? Is it catalyzing reaction? Is it a building block? Is it making membranes? So there are a lot of different functionalities, and they're all linked to the three-dimensional structure of the protein and what it can do with different binding partners and other proteins in a biological environment. So how does this happen? We have an unfolded amino acid sequence that was basically read from the DNA, then RNA, and then we have the sequence of amino acids into an unfolded chain, and somehow this will fold into its native structure. Now, one question could be, how do you get from the unfolded amino acid sequence to the actually folded state? And here, because you have a lot of different degrees of freedom, this is a very flexible chain, you also have a huge number of possibilities to get there. So the pathway of how to get from the unfolded to the folded state is something that is relevant to know, and I will get back to this towards the end. But then it is also very complicated. So what we will see in the rest of the lecture is, can you just jump over this and not try to understand why and how, but just the end result. So we know the chain, and then we know how it should be folded in the end, only at input and output. For decades, to understand protein structure, you need to have some experimental structures, right? So in practice, you would do experiments, but of course they are very expensive, time-consuming, and the experiments that you might know here that are also part of the protein database are coming from X-ray crystallography, nuclear magnetic resonance, and in the past decade, more and more cryo-electron microscopy. So this is then the structures that have been, over decades, inputted into the protein data bank. So we had a large set of different proteins, but they were not always accessible. And if you wanted to study a disease or a phenomena and you need the protein structure, maybe it was not available, so you would have to go to computational models of, just to model reality and have a starting point to understand what's going on. And the state of the art is AlphaFold for computational structures. But what did we have before this time? So before 2020, before the first version of AlphaFold came in, which already utilized AI, there were two different ways for protein structure prediction. One was really template-based modeling. So you need a known protein structure. You have the sequence of the protein of which structure you would know. You can make an overlay of them and try to go there. Or if there was no template, so there was nothing closely familiar in the protein database, you would have to do it from scratch and maybe use template-free modeling. And what you see here basically is that if you had a good template that you can put your amino acid on and get a structure, you might expect some higher accuracy. But if you had no idea of how the structure would look like, there are no homologs, no related proteins, then the model accuracy might be quite low. And in the end, it is, even with AlphaFold, it is always a model, so you always want to have an experiment, but how well can you get the protein structure directly from the sequence? So the entire idea also with AlphaFold is more on the template-based field. So as I said, you would have a known structure, and also you know its sequence, and then we have a new sequence, and what we can do is an alignment. And then you could see, okay, how close are the sequences together, right? And if the sequence identity was high enough, then you were also confident that the homology model that you would build on the existing experimental structure would be quite good. And that means once you had your computational structure, you could say, okay, now I can go and do further analysis using molecular dynamics. Depending on which protein you're looking at, you might want to do drug docking or anything that's linked to your actual research question. So what is really important in these computational approaches or in any kind of methodologies is how good is my method performing? So you can have, this is the basic principle, but how are you making this alignment? There might be different techniques to do this. And a nice way in protein structure prediction, like the golden standard in the community, is the CASP competition. So this is also called critical assessment of structure prediction, or you will also find critical assessment of protein structure prediction. But the idea is here that you have a blind test, a challenge open to any computational groups who are making protein structure predictions. And the way this is built is that there is a crystal structure, experimental data, that is not yet published. And then you just gave the sequence of that specific protein out to the groups, and they were supposed to predict the three-dimensional structure. And in the end, they handed that into the CASP team, and they evaluated all the theoretical models that came in with the experimental structure that was released. And then they published the results, and you could see, okay, this is the group that was best performing. And for years, this is a very challenging problem, like how do you get from sequence to there? And before there was any AI, you can see this in the next, so this would be kind of a prediction, right? So in blue you have, in green you have the actual experimental structure, and then in blue you would just overlay the computational model, and then you can see how far are you off by some specific metric, like root mean square deviation of the alpha backbone, or there are different metrics that you can look at. And so if we look at the best performing methods over the years for this competition, you see that it was not that great. So 90 here is a score where you can say this is getting really close to experimental accuracy. So this would be a very good model prediction. And until 16, it was around 40, with ups and downs. I mean, there are also easy structures that came out, also more challenging protein structures. I mean, even from the side of the protein structure that is handed out for this context, there are different degrees of difficulties. But then in 2018 came AlphaFold 1, which was the first method that started using AI. And then there was already a jump in prediction accuracy. I was then, during the competition in 2020, which make a huge jump, and I'm going to talk today a bit more about this AlphaFold 2, this is really the AI model prediction tool that made the news, where people said, oh, the code to protein structure prediction has been solved, and now we actually have protein structures that we can directly use. So this is a bit the rise of AI in this competition. And then the question is, okay, what is happening next? So how did it get to this, and what is the impact and I would say opportunities and challenges that you have now in this field of structural biology, molecular biology, and all the surrounding fields? So here again, what is happening? What the model was trained on is basically sequence. You can have sequence information, so you want to go from the sequence, you want to input it into your model, and you want the 3D structure. And in this case, this is going to be AlphaFold 2. And what is happening exactly in this model is important at multiple levels. So what is the kind of architecture, what is the idea behind, but how far can we actually look into this and what it is doing? Because we have heard before that this is how neural networks work in their fundament, but you have seen that there are many hidden layers. And also an AI like AlphaFold has a lot of layers. How can you understand what is happening? So this is going to be a challenge. But if you look under the hood here, this is what you find in the publication that was published in Nature 2021 by Jumper et al. And so this is a higher level workflow. So there are more details in the different bits, but at a higher level, this is what is happening. And you could simplify this in 3 different building blocks of, say, what is happening. The first one is taking input from databases and, we could say, data pre-processing, using evolutionary information and also pairwise interaction matrices. The next one is what they call in the paper the evoformer. So there is, once they gather all this information, there will be cross-communication between the evolutionary information and, let's say, this pairwise matrix, which gives them an idea of how the different amino acids in the protein are interconnected. And in the end, there is a structure module that finally yields the 3D structure. So herein you have different constraints from, let's say, physical chemistry or like specific geometry constraint from amino acids and from the backbone that goes in. So what is happening in the first part, and what was so important for this? So one of the ingredients for AlphaFold is this, it's going to be this genetic databases. That means that you can basically use information from multiple sequence alignment from different organisms. So if you have your sequence, this is a target sequence, you can still find information about related proteins from different organisms. And what is interesting about this multiple sequence alignment is, are there parts of the protein that are conserved, so that you find them in all of them? For example, this position looks like it's all the same, so probably this amino acid is important for the function of the protein. It has been conserved in all the species. There might be some that are completely different. So they are mutated and they don't seem to impact protein function. So you get information from the evolutionary development of the protein here. And then from the structural database search, you can also get an idea of the pair representation for your amino acids. So you look at all of the amino acids in the sequence, and you can look at some metric, for instance, the distance between them all over. But for here, you actually need structural input, right? So, and in that case, you would maybe, for the model at the beginning, just take an initial guess of how this could look like. So this would be the first part, and these two are going, there should be an arrow here, so these two are going to be, and this is one important part of this evoformer, is that they are communicating back and forth. So if you look into the supporting information of this paper, then they show in depth how often this actually happens. And the idea in the end is that this is basically updated. So you used the information of the different sequences. You also look at the pairwise representation, and in the end, they come up with an updated, with the sequence and an updated pair representation, which was fine-tuned by that model. And then you can come to the third block. So with this output from the evoformer, it goes into the structural model module block. And what happens here basically is that this is yet another neural network. So the evoformer is a neural network architecture. Here's a second neural network, which then performs different operations on the actual protein. So like translations, rotations, and then there are physical constraints that amino acids are represented as triangles, and you kind of fix the distances between them so that you have basically sensical distances while the model is learning how to build the structure model. And the end result of this is then to get a 3D protein structure. And you also get a metric. So I don't know if any one of you have used AlphaFold for structural prediction, but in the end you have your 3D protein structure, and you get a metric to identify regions of high and low confidence in the model. And so this was, if you look at the performance that AlphaFold had in the CASP competition, this was a high breakthrough. And yeah, this was very important, or this is very important, because before you had you had sequence, and also in the time of omics, we have more data available for different organisms, but we didn't have the direct link to structure. And so once they had this, they also started to really build the structure for the entire human proteome. That means that now there are actually protein structures. You need to be aware that they are computational models, but it's a very good starting point for all the downstream analysis that you're doing afterwards. And thinking of the impact of AlphaFold, it was a real breakthrough in protein structure prediction, or it is really. It also accelerated scientific research in different areas, because now you had structures. You could go for looking at drug binding sites. If you had another machine learning model where you were only using sequence, now you could actually check, if I feed it structural information, do I get a better prediction? For instance, for protein hydrophobicity, or for something that is disease-related. It's kind of a huge jump up. Application, yes, so drug design, enzyme engineering, structural biology, and indeed linking omics data to structure, because if you think of the dogma in biology, it is DNA, RNA, and then the protein structure. So if you have everything in there, you have genomics, transcriptomics, you have, then you can also link it to the actual 3D structure of these proteins. And then I just checked out of curiosity, I hadn't done that before preparing for this talk, how often has it been cited? And since 2020, it has already been cited over 25,000 times. And it is really being actively used. So of course, you have citations which just mention it in the introduction or in the conclusion as a nice outlook, but no, people are really using these structures. So this has been really a huge leap forward. But then with this come also different questions. So we said that these neural networks are kind of a black box. Is this really what you want? Or what is the research question you're asking? So I think it's very important to have these structures. But as I've said in the beginning, how is this happening? So in the AlphaFold paper, you see that there is a claim saying that this uses now basically physical chemistry and physics in building the model. Really, there is structural information. There's also this pairwise matrix, but there is little physics in the sense that there is no thermodynamics, there is no hydrophobicity gain in that model. So you do not learn anything about the protein folding process, right? So what do we know about how the protein is folding, this is not something we can get out of AlphaFold. And once you have your prediction, you can always go back to the model and then change the data set or change the training parameters and try to see, do I make the prediction better or worse? And you can understand which ingredients in the model were important to get the good prediction. But this is still a very limited view. It's not really opening the box and saying, oh, this is why it folded this way. So there's still a big gap between the prediction, even if it's a very good prediction, to understanding how we got there. So it's a bit the same that we had from your question in the last talk. It's exactly touching upon this. And also, what are the limitations? So it was trained on the PDB data, available PDB database. So what if there is something that is completely new? Or how about intrinsically disordered protein? What happens there? How good would be the model predictions in such cases? And so if we think of protein folding, why is it, you could say, maybe you don't care about how proteins fold because you know the end result. But this is not quite true because biology is complex and proteins might also fold differently depending on the environment. If you think of alpha-synuclein, it will have a very different folding behavior based if it's on its own, close to a membrane. So this would be a tricky kind of a chameleon protein. But if you think of complex processes such as neurodegenerative diseases like Alzheimer, Parkinson, then something goes wrong. You have nicely folded proteins that perform their function, but then something goes wrong. So what happens normally is that these proteins have a high number of hydrophobic residues. So that means hydrophobic residues are residues that don't mix with water. So the way we understand the protein folding process at the moment is that it is due to the hydrophobic effect. That means if you have a protein sequence, there are hydrophobic residues, and they're surrounded by water, and the idea is that by collapsing together and burying the hydrophobic residues into the inside of the protein and keeping hydrophilic residues outside, you get into some kind of a protein folding shape. This is a rough idea of how things could go. What happens in unfolding is that this protein would partially unfold, opening up to these hydrophobic patches or hydrophobic residues. And what happens then is that it can easily get in contact with another protein and then start aggregating, right? And then it would form these toxic aggregates and very stable amyloid fibrils. So they are structurally very stable once they start to form. And how this happens exactly, so I've really simplified it here, but if you look into current literature, what people accept for this mechanism is first you have these monomers, then you have toxic tetramers, and then you have another aggregation of these, and then so it's a very complex figure with different pathways that people draw here. So how this is happening, this is something we don't know, but this is something that would be very interesting for drug targets, for really understanding what's going on here. And then the other thing which is funny, so for AlphaFold is you get sometimes structures where the model, so for example, if you gave it this sequence, I think here's the same, the blue is the model and green is the experimental structure. This is only one part of the protein, so it's a trimer, but the model still recognized that this is the structure, how it would be, even though it wasn't actually given the stoichiometry. So they didn't input that there should be 3 protein chains close to each other. So this was one of the interesting cases that they also discuss in the Jumper paper. So there are still really open questions here that you need to that would need to be checked. But the nice thing about what I like about AlphaFold, also about this Nobel Prize, is that it really shows a great example of a complex problem that was tackled by our interdisciplinary team. And I think this is exactly what is needed for very challenging problems. So it's a bit, again, like we said before, these are not like isolated categories, everyone doing something by themselves, but these are teams of people working together. And it's very interesting to work with a biologist, a computer scientist, chemical engineer, physicist, because they have different point of views, and in the end product is the process is a lot of fun, but also the results can be great. The other thing is that AlphaFold would not have been possible without decades of effort from all the people who, took in the work to build the PDB database, to make these structures openly accessible so that they could really train on it. And the other thing that cannot be underestimated is the importance of these challenges or blind challenges like CAS, because this is kind of setting a golden standard and also to make a fair benchmark where you can challenge yourself and compete with colleagues for a specific methodology. And it's also a possibility to then benchmark and assess the quality of given methodologies and then improve it for the next round. And then, of course, there is the developments in AI. So there's a huge increase in neural network architectures, which were used here, which are also have a huge impact in physics, but also in other fields. So you can see these developments are very quick. And I think it's very exciting times now to see what you can do with deep learning for these kind of biology things. Now, very briefly, because I said I would focus on the protein structure prediction part. Based on this, also having now all these structures, the next kind of imminent step would be, okay, is there a way to now get to de novo design? Like you want to develop new proteins. Of course, you also need their structure because structure is linked to function, but you would like to go more to decide molecular properties, maybe find new therapeutics if you have a specific drug target, what kind of polypeptide could link in. Can you fine-tune molecular targets from scratch? And this can be for proteins, but also for small molecules. And very brief, this is part of a picture from one of David Baker's publication. But what they use here is technology very similar to this image generating AI that you might have seen like DALL-E. And just to give an idea of what's underlying this is basically noising and denoising process that they're doing. So under the hood, they just use protein structures, and then in the forward process, you kind of add noise to these structures until you get some random noise distribution. And the nice thing about this is you need protein structure as your starting point, and the noise you're adding is controlled. So there you have really full power of how you're doing this. And then you train a model to do exactly the different process, to go to the denosing and try to learn it how to get between the different steps. And this is, in a nutshell, a bit the idea of how you could then start generating de novo proteins from structure. And this is falls under the hood under this generative AI processes. I think this was kind of a similar flow, starting from this AlphaFold and the deep learning methodologies that are coming up. And there will be, I think, many more possibilities here, because it's not only for proteins, but for small molecules. So in terms of therapeutics, also in terms of other materials. And this is a very interesting technologies that you can see here. So where would the next revolution be? I think using deep learning to continue for this de novo molecular design is very interesting. After AlphaFold, Google DeepMind also looked into mutation impact prediction. So if you already have protein structures or you know the protein, you changed one single amino acid residue, what is the impact on the protein's function? And can you link this back to specific diseases? The next really important part, I think, mainly the explainability of the models, and then also how you can make this fair and open access, that people can also use it and continue to build on the available work. And for the explainability, the thing is that you really want to understand why the model made specific choices. So ideally, it should not only be about prediction, but you also want to understand the underlying biological process. So why is this going? And what people are now doing a lot is also starting to use AI not only for a prediction task, but you can use it to make sense of data that you have. So basically, you would use AI methodologies to understand your biological data and maybe see, okay, if I have genomics data, for specific cancer types, then I can actually see what is happening at a molecular level, which genes are involved, and I can make more sense of the data that is available out there. Yeah, and with this, I would like to stop or to end the talk with the Nobel Prize winners and the topic for which they were handed the prize. Thank you.
Wonderful. I'm sure there's some questions in the audience. Yes.
Thanks, very nice presentation. Something that an AI does very well is dependent on the context, say, now I'm going to flip from turning right to turning left. And that actually is for something, for a field like protein folding, where this also happens, right? Protein always folds properly, and then some context change happens, and before you know it, you've got Jacob-Creutzfeldt or something like that. These flipping points in the biochemical reality, I think, are very interesting to train into an AI and to make sure that the flipping point comes at a certain place. But when you give it only spots, this folds like this folds like that, this folds like this, the AI has to guess where the boundaries are. Is there any work going on to training the AI explicitly with those boundaries?
Good question. I'm not sure there. No, but yeah, okay. No, but I see the point. So that would be still, you could still benefit from actually understanding why a bit more the actual folding process. Yeah, I mean, if you look at the current state of protein folding, there are different models to do this, but I mean, they're far away from the actual understanding of how to completely fold the protein. So maybe you could use some preliminary steps in there, but yeah, which one to use? I'm thinking, yeah.
Okay. Come on.
Yeah, thanks for the great talk. Do you have any idea how the complexity of this problem compares to maybe this ChatGPT or so, which is billions and billions of parameters? Do you have any idea?
You mean from the size of the model?
Yeah,
or?
yeah. Or time it takes to do this, or?
I think ChatGPT is way heavier, right? I mean, this is the data that went in. Also, the parameter size of that model is for you, but I'm not sure exactly like how many % the AlphaFold model would use in this. But that's a good point. This links a bit to the last bit that you were saying, right? How much energy goes in. Yeah, this is also something that people don't really talk about a lot, right? You build these new AI models, and now you also see there's ChatGPT, it's in English, and then there's a lot of other initiatives in different languages. It's very expensive, and it's very energy consuming. So you at some point might have to think, do you need an AI model? It's also just in normal bioinformatics, like normal, like say master students projects or so. It's very exciting to hear AI, but sometimes it's just, you don't need it. And if you don't need it, then you shouldn't use it. So it's really about the problem at hand, the research questions, and if AI is the best method to get there. But I think the nice thing about this protein structure determination problem is this is actually a very nice case where you could see, yes, here you had a huge benefit from AI, right? It was always a little bit in the same direction. Then you have already in AlphaFold 1 neural network, which was quite simple, and there is a jump. But then when they fine-tune it with the Voformer and the structural input, then, so here, yeah.
Thanks very much for a nice talk. My first question was also about the computational complexity. How long does it take to actually calculate this? And can everybody do this on your own laptop, or you need some server?
You mean now getting, like if you have a sequence and you want your protein structure, you can do this on the server. Also, there's an AlphaFold.
And is it a commercial product, or is it open to everybody?
Yes.
Commercial.
Right, and sorry?
Yes, it is commercial? Or
No, yes, it's open, so you can get
nice.
the structure. The structures easily, and there are also now databases where this has already been done, so you can directly get the structure models.
Thanks.
So this is very useful.
Thank you. It was a great talk. If I see these protein structures, to me, they always look like a bit floppy and deformable. Is that a wrong impression, but you are already nodding, so it seems to be correct. But then
Yeah,
how does this system deal with this kind of,
yeah, this
deformability?
This is a bit the same thing that you have. So you don't have protein, you don't have dynamics in here, right? It's a fixed model. It's the same when you look into the actual experimental structures, like X-ray crystallography. So this is a fixed point of the protein structure, but it's actually a wrong image of a protein, because proteins are dynamics, and they're also in a crowded environment. So there are a lot of other proteins around. There is water. It's very flexible. So even before, if you looked at X-ray crystallography structure, you cannot be sure if it's the actual functional biological structure of that protein, because the conformational space is much bigger. But it's a structure. Yeah, not even necessarily. It might really be a structure on in a local minima. Maybe the active function is actually in another minima. And ideally, if you have molecular dynamics, you would hope to be able to sample between the different potential wells, but that's not necessarily the case. So if it's a deep well, then you might actually be stuck there.
The second question over here.
Yeah, as someone who's trained in chemistry and now like doing research, I'm really curious because from Wilfred's talk also we heard that these models have been around actually for quite some time already. Is there a reason why they are now rolling out, or should more people, next from studying some hard science, also study machine learning at the same time to better understand this is a tool you can use for these types of problems?
Okay, so I think the reason why they more used now is you have computational power, you have more data. I think everything is timely coming together for these kind of approaches and technologies to rise. And then there are really active people, very active in the development and fine-tuning of these methodologies. For, I think, life scientists, or let's say life scientists, so biology, chemistry, physics, chemical engineering, not everyone can develop AI and always use them, but I think it's important to understand the language, to be able to see, okay, is this an AI problem? Would that actually be helpful? But also to be able to say, okay, there is an AI model, but this is great for this bit, but there are also limitations. Or it's really bad because there is a lot coming up. So I think doing an introduction to AI, and there are a lot of good courses online. Universities usually have one, and then you can basically have the practicals are then fine-tuned on bio or chemistry data, which is nice. But otherwise, doing one of these introductory courses on either machine learning or machine learning and deep learning, I think it's worth investing just to be able to follow these discussions.
Over here. Very nice talk. You were referring to, or my neighbor was referring to the floppy structure of a protein, right? This is, of course, the secondary structure, only what you show. When you go to the tertiary structure, which really determines the structure of a protein, does the AI also take that into account?
Well, it's a tertiary structure that you get, right? You get the 3D structure in the end. But, I mean, this is also...
From the primary structure, you go to the secondary structure, which is the alpha helixes and the beta sheets. But in the end, of course, it's a space-filling model, which is very dense.
Yeah, but what you get is really the tertiary, right? So you get the right.
Yeah, but there are interactions between one beta sheet and the other, and that's not from the structure, from the primary structure itself, but it's from the secondary structure, which then interacts. Yeah, the parts interact, and does the method also take that into account?
Yeah, it's really because it's trained on the PDB, right? So on the finished 3D tertiary structures, right? So this is the information it already has, so it can get this over. So this, yeah.
Yep.
Thank you for your talk again. Again, I have a question. So, I'm also studied in physics, so I would like to ask: actually, the essence of protein folding isn't an energy minimization problem? Because John Jumper's studies, by the way, Professor Wiel, John Jumper got his PhD on 2017. He's very young, I guess. Isn't it in the physical sense like an energy minimization problem. As you said, also like trapping the hydrophobics into the center so that you can create a mycelial structure, basically, than you get from the lipids.
Yeah, you have the potential energy surface, right? And you have but it's a very complex surface, right? So it's sometimes simplified, like with a few minima, but in the end, it's a huge. It's a huge landscape, right? And also, you're going from completely unfolded to something that's completely folded. So 1 cc bond, right? You can rotate it 3 times, and if you have different, different substituents, you also will get 3 different energy minima. This is just for one bond, and here you have the entire backbone. So this is also why protein folding cannot be an arbitrary. So it's not going to, visit all the possible configurations until it finds the right folding space. It's really a driven pathway that's going from fully stretched to the energy thing. But here the model is, based on these structures, able to go from this. It's really sequence to structure immediately.
Thank you for this wonderful talk. I had a question about, you showed that the model has uncertainty, and this is a very important part in machine learning. Can you tell us a bit how does this work, this uncertainty? You also had a nice picture where you said, there in the protein, I'm not certain. You mean
exactly. the confidence of this? Yes. So with the structure comes also a confidence score of how confident the model is that this is a proper structure. And then if you go to regions, this is here, for example, these loop regions, they're highly flexible, right? And sometimes in the PDB, in crystal structures, they're not even in, because they were so flexible that you cannot determine exactly where the position is using X-ray, so they're missing. And this is challenging. This is also a problem for intrinsically disordered proteins. So there, it's still a model, right? In the end. So this is something that you have to be aware of when you use these structures. That these are predicted structure models. For exotic proteins, completely new protein family, that might not be right. And then you would have to check what. So in the PDB you have X-ray, you also have NMR. So in the NMR structures there is more of the dynamic, because there you have basically ensembles of proteins, right? It's a different type of structural determination method, but it's only for parts of proteins, only for small proteins. So you don't have this for the larger proteins. You don't have this for membrane proteins, which have some other construct linked to them. So there is definitely uncertainty.
Maybe a final question over here.
My question is more related to like the later part of the model, like the actual protein structures, because as I understand it, the model only gives you the structure, and then your talk on an early stage you told how closely related the structure is to the biological functioning. How can machine learning actually help kind of determining the biological function of a protein structure by kind of comparing it to other protein structures?
So now you want function prediction. Yeah. This is something that is coming up in a lot of projects. So I'm now starting with some people on a project where you have like you have all of this omics data, and then the idea there is to identify specific enzymes that cover off flavors in. For example, in vegan cheese or so, and there is the same. You have all these proteins or genes, and you want to identify which of them are actually enzymes. There are AI models for this, but it's not that easy. So I don't think we're at the point where you can just give in sequence and then say, okay, this is the structure. But based on the databases where we have the sequence, we know the function. Then models are trained, and you try to also with a high confidence, ideally, be able to predict the function of that protein. But this is definitely work in progress.
I think the conclusion is that there's still a lot of work to be done, and that AI is not taking over our jobs. For the young people, please join these fields and work at it, and with a lot of benefit. Let's thank Halima once more for a beautiful presentation. Also our first speaker, Wilfred. I think it was a wonderful evening.